Hey, everybody, we're just going to get started in just a few minutes. Just waiting for Dr. Blanar to log in. In the meantime, if anybody has any questions or anything, there is a Q&A section as well as a chat as well we can engage with. And then at the end, we'll have the opportunity to open up some questions to the audience. Chris, are you able to get in now as the co-host? Oh, there he is.
And now I've unmuted myself, so I think we should be good.
All right, I don't know what happened, but I'm glad you're here.
As am I.
Well, I just want people are just coming in right now, but I think it's 6 o'clock, so we can get rolling. We've got a good crowd that's here tonight. One of the things I do want to just give everybody a heads up who's attending. There is a Q&A section here. This is set up as a Zoom webinar. There's also a chat box as well. So Dr. Blanar, I'll be moderating tonight's session in here. So we'll probably let you go through your presentation unless you want to take questions immediately from the audience. And then at the end there, we usually leave about 10 to 15 minutes to answer some of the questions in the chat box, as well as open it up to questions from the audience as well. So with that said, I just want to introduce everybody here to Dr. Blanar. So tonight, basically, this is a virtual lecture brought to you by the Halmos College of Arts and Sciences as part of our fall virtual lecture series. My name is Matthew Adams. I'm one of the admissions directors here at the Halmos College of Arts and Sciences. But I'd like to introduce Dr. Blanar. He's an associate professor for NSU's Department of Biological Sciences, and he has taught undergraduate and graduate courses here at NSU, from biology to epidemiology, parasitology, and more. Some of Dr. Blanar's research interests include you have to correct me on this, parasitism in coastal habitats, parasites and migratory behavior in Everglades fish, biocontrol of parasitic nematodes, is that how you say it? Used in engineered bacteria and parasite community ecology. But he's written and co-authored numerous articles and presentations on the topics. And tonight, he'll be basically a large part doing an informational webinar on ventures in parasitism, hosts, parasites, and other unexpected relationships. Myself and Dr. Blanar will be able to answer any questions you have regarding any of our graduate programs or his lecture tonight. So at the end, like I said, about the last 10 to 15 minutes, we'll open up to the audience. All right. And with that said, Dr. Blanar, take it away.
All right. Thank you very much, Matt. Just making sure that you can hear me clearly. Is that good?
Sound good on my end? Anybody else? Just put it in the chat and let us know if you can't hear.
All right. Give me a moment. I'm just going to start the presentation and then share my screen. All right. Here we go. Okay. Just make it like this. All right. Can you see the whole presentation here?
Yep.
Cool. All right. And can you still see my face on the screen? I'm going to take that as a yes. Okay, folks, thank you very much for joining. And I am very, very pleased to be giving this this talk. I had a few options about how to do this. And what I decided to do was talk about my own pathway. I've never really done a talk like that before. And so this gave me a chance to talk about my adventures in parasitism and talk about some of the unexpected directions that my path took me in. I understand that many of the attendees to this seminar are people who are just really at the early stages of figuring out what they want to do. And so it seemed to me that this would be a good opportunity to talk about my own path and to hopefully help you get some feedback on how these paths can be non-linear. I definitely refer to my own path as non-linear, and to hopefully give you some insights on how I chose my path, which is I didn't really, and talk about some of the interesting adventures I've had along the way. So it's going to be non-linear, but linear-ish, mostly in chronological order, but kind of not. So bear with me, and hopefully, I think that there's a payoff at the end. So first of all, some background on myself. I really don't want to go over what Matt went over in his great introduction, just to talk about my own path. So I did my Bachelor of Science split between McGill University and Concordia University in Montreal with a specialization in ecology. And after that, I continued and did a master's of natural resource sciences at McGill University at their agricultural campus on the western part of the island of Montreal. And then from there, I moved east to the east coast of Canada, to St. John, New Brunswick, and I did a PhD in biology at the University of New Brunswick at St. John. And then after that, I did 3 years as a National Sciences and Engineering Research Council industrial postdoctoral fellow so I was at Environment Canada for that. And the NSERC is Canada's NIH, if you want to compare the two. That's roughly the Canadian equivalent. And so, when you present it this way, the path looks pretty linear. But what I want to do is to highlight the fact that nothing about my career has been obvious. Nothing about my career has been linear. And in fact, it always makes me laugh to sort of go back and look at the pictures from these various eras, because what strikes me the most looking at these pictures is I definitely do not look like a guy with a plan. I look like a guy who's following the path of most interest and the most fun. And looking back, I would have to say that was the strategy I took, and it largely panned out for me. Either I'm really good or I'm unbelievably lucky, and I suspect it's the latter that helped pull me through. So despite all of the sort of no plannedness, I do want to talk to you a little bit about the pathway, because being wrong multiple times really helped me out. And I think that all of this at the end is going to come together in some coherent piece of advice that I'll be able to give those of you who are just starting out, whether you're just starting out as an undergraduate, or whether you're starting out as a graduate, or even if perhaps if you're an early career person, I'm sort of wondering what these paths should or shouldn't look like. Maybe I'll have a message at the end of this for you. So the first thing I want to do is talk a little bit about my time at McGill University, and specifically talk about the key role of mentorship. One of the most important things you can get out of your studies is finding a mentor that you get along with and that you bond with, and that you and that teaches you important lessons, and not just the scientific ones, but the real life ones. And I was lucky during my undergrad to have 2 such mentors, and I'll talk about them right now. The very first one is Robert H. Peters. So Robert H. Peters was a big guy in ecology, and he had written classic texts that are still used to this day in ecology. He wrote The Ecological Implications of Body Size and another book called for Ecology, which I still read probably about once every 2 years now, because it still has important lessons and it reminds me of important things that I need to keep in mind for my own work. But Rob Peters, for all of his importance in the field and so on, was an incredibly human guy, an incredibly pleasant person to talk to. And I spent hours in his office just talking about science and the practice of science. And he taught me a couple of really important lessons. The first lesson probably being the most important, which is the more you like a theory, the more likely it's probably going to end up being wrong. In other words, the more attached you are to a particular hypothesis, the more likely it is to get shot down. The other lesson he taught, which I really liked, was that just because the story is really good doesn't mean that it's true. And he warned me that ecology was full of these sort of things that everyone just thought they understood, that were what people sometimes call just so stories. And he trained me to sort of recognize when those stories are too good to be true, and to dig down and to actually see if this thing that everyone, quote unquote, knows is true is actually supported by evidence, as opposed to just a really good story. Unfortunately, Robert Peters passed away of cancer back in the 90s, and so he's lost to us now. But his books are still out there, and his papers are still out there. And if you ever want to read really interesting, really thought-provoking books on ecology and the practice of science in general, I strongly recommend both of these books. So he was my first mentor at McGill. I was very fortunate to have been there when he was still around. My second mentor was a crazy guy called Joe Rasmussen, who I met on a field trip at McGill's field station. And he had two students with him who are on this image too. The one in the middle is a guy called Gilbert Cabana, who is a professor now at the University of Trois-Rivières. And the guy on the right is a guy called Paul White, who's at the University of Ottawa. And these three guys together, but especially Joe, played a similar role to what Rob Peters did for me. And they talked to me about the practice of science, and they told me how to walk away from certain problems and so on. But one of the things they had was boundless curiosity and energy. And these are the guys who introduced me to parasitology. So I was swimming in a lake with Joe and Gilbert, and we found these fish, and these fish had these black marks all over them. And I was like, what is this? And they said, oh, it's a parasite called black spot. And we started sort of asking questions about this, and they encouraged me to do an undergraduate research project on that subject. And that's what I did. And so those of you who are interested in and who are still at the undergraduate level, the lesson here is that if there's any way at all that you can sign on to an undergraduate research project, I strongly recommend it, because for me, it was this moment where I realized that it wasn't just, you didn't just need to learn by reading books or by reading papers or by talking to people. You could actually learn by doing the science yourself. And if you had a question, there was actually a process that you could follow, and you could actually learn to answer questions for yourself. And so doing that project got me into their lab, got me participating in projects, and it was by far the biggest stepping stone that got me into doing science. And the reason why I'm a scientist today was because of guys like Joe and Gilbert and Paul, who talked me through the process of doing research and guiding me. I also learned about failure early on, thanks to those guys. So the particular system that they had me working on was black spot disease in fish. And in fact, it was in pike. And the lake that we were studying was this small lake at the McGill field station. And these fish were, this isn't a fish from that particular place, but it was exactly as bad. You could see that these fish are covered in these little black dots. Almost looks like they were sprinkled with pepper. Every single one of these black dots is a little encysted parasite. The species that causes this is a parasite called Uvulifer ambloplitis. And the adult version of the parasite lives in birds like kingfishers and things like that. And then it goes through snails, and then from the snail, it goes into the fish. And the bird gets it when it eats a fish that's infected with this thing. And the population of fish that we were studying were super, super small. Like they were super skinny. Like these were talking pike that were maybe this big. And if you know anything about pike, you'll know that they can get this big. They could get absolutely giant. But none of the adult pike that we were looking at were longer than maybe a foot and a half long. They were super, super small, and they were riddled with these parasites. And so my question was, is it the parasite that is stunting the growth of these fishes? So those three guys, Joe and Paul and Gilbert, helped me design a project, and I spent part of my undergrad collecting data for that project and analyzing it. And I got my first taste of failure when we realized the parasite had absolutely nothing to do with it, because there were adjacent populations that had the parasite that weren't stunted, and these fish here, if you raise the young away from the parasite, they grew just they were still kind of stunted and so on. So the whole idea of my undergraduate research project didn't pan out. It was what we call a negative result. In other words, I had a hypothesis: the parasite was causing this problem and stunting their growth. But the hypothesis was not supported by the data, and it was on multiple levels a good introduction to the practice of science, and like I said, an early taste of healthy failure. So from there, I got the parasite bug, and I didn't necessarily set myself up to become a parasitologist, but it just so happened that the next opportunity that happened was in the lab of a guy called Mark Curtis, who's retired now. But he was at McGill University, and he studied Arctic fishes, parasitism in Arctic fishes. And so he got me turned on to this project that was being financially supported by the northern indigenous peoples of Canada, the Inuit. So we call them First Nations. And so the northern First Nations in the part that I traveled to, they call themselves the Inuit. And so I got to travel up to a town called Igloolik, up in the northern, up in the Canadian Arctic. They have a really cool research station, which looks like a spaceship, which you see in the image here. I got to collect a whole bunch of fish that were infected with a parasite called Diphyllobothrium dendriticum, which is a fish tapeworm. And so here, this is a fish that's been opened up, and these are the guts of the fish, but all of these white pimple-like marks, this is a juvenile stage of this tapeworm called the plerocercoid. And the adult version of this parasite is here. And the Inuit essentially hired or funded this project and hired me to do this research because they were concerned that this parasite was negatively affecting the health of the fish, and also that it was making them more susceptible to pollutants that were up in the Arctic. And so they hired me to do this project. And so as part of this project, we had to recreate the life cycle of this parasite in the lab. And so we figured out how to put these things into the first host of the life cycle of this parasite, which is a little shrimpy type thing called a copepod, a little arthropod. And here you can see the plerocercoid, so the procercoid of this parasite, the first larval stage of this, or one of the larval stages of this parasite in this copepod that we artificially or that we intentionally infected. And then we could then feed these copepods to fish and then do this work experimentally. And I said that my undergraduate experience gave me a first taste of failure. This was a second taste of semi-failure. We had a huge success in that we were among the first to recreate this life cycle in the lab. And so we were able to study it and learn some interesting new things about it. But the specific project I was hired to do, our hypothesis ended up getting shot down. So our idea was that the parasite and the contaminants that were present in the Arctic together were combining to cause additional sort of negative health impacts on the fish. And we found really just no evidence for it. I mean, what evidence we did have was pretty weak. And so it was a good idea. And we didn't actually really find a super strong effect of the parasite on the fish, all right, which is a bit of a running theme here. But you can see all these parasites. This fish would have lived out its life probably and been perfectly happy, even with all of these larval tapeworms tunneling their way and making little cysts on their viscera and stuff like that. But again, it was another taste of parasitology. It was a different system. It got me up to the Canadian Arctic for about 8 weeks, for which I'll be forever grateful, because it was, again, another sort of life-changing experience. So after my master's, I decided that I was going to take a break from parasitology and maybe work in a different system. And the universe had other plans for me. And as it turns out, the very next project that I did ended up involving parasitism as well. So I said earlier that I followed the path of most fun, but I also kind of followed the path of least resistance, and that path kept leading me back into parasitology. So most fun and least resistance. I'll take both of those options. Thank you very much. And that led me to becoming a parasitologist. So this brings me to my time at the University of New Brunswick doing my PhD, and I did that under two mentors, Deb McClatchy, who is now the president, I think, of Wilfrid Laurier University in Canada. So she has reached the absolute pinnacle of the academic scala naturae. Like, she's absolutely at the top now. It's impossible to get higher than president, I think. And so Deb is now the president of a Canadian university, a major Canadian university, and it's fantastic for her. And so she was the one advisor, and then the other one was a guy called Kelly Munkittrick, and both of them played incredibly important roles in shaping how I think about parasites and so on. Deb had absolutely the best advice for me. Her thing, anytime I came to her with a cool result, she would look at me for about a minute. And she wasn't a parasitologist, so she would look at me with a kind of disgust, and then she would go, "Who cares?" And she didn't mean it aggressively or meanly. She was like, what she was saying to me is, she's like, "I'm not a parasitologist. You're coming in here with all this nerd language that's parasitology specific. Make me understand why this result is interesting or fun or informative." So then I would have to sort of explain to her, like, this is why I think this result is cool. And so she meant it. She meant it very well. And we ended up having great interactions. And she was absolutely fantastic as a supervisor. And then the other one, the other guy, Kelly Munkittrick, taught me about the how do I say this, like the practical application of science. And he taught me about making your science meaningful and making sure that your science addresses real world problems and so on. And so his big question to me was always, is it real? And so with Deb, it was, who cares? And with Kelly, it was always, is it real? You're looking at this pattern. Is this is this pattern statistically significant? And even if it is statistically significant, is it biologically meaningful? And if so, how is it changing the ecosystem, and where do you go from here? And so they were absolutely fantastic mentors. Again, another fantastic project working on another fantastic parasitological system, this time with a gillworm called Disco cotyli sagittata that infects the gill tissue of salmonids, so salmon and trout and things like that. And so here's the parasite. These long skinny things are the gill filaments of the fish. And then this parasite here, this is the parasite. You can see the clamps that it uses to hang on to the gills. And you can see its eggs. It's a beautiful, beautiful parasite to work with. And my research project was absolutely fantastic. I got to go up into the Canadian wilderness, into rivers with a backpack that shot electricity into the water. And that allowed us to get fish. And then we took those fish and anesthetized them and measured them and weighed them. And then I got to examine them for gillworm. It was possible to do this without killing the fish. And then we would release the fish and they would recover. And so we had all kinds of interesting data about this gillworm and its distribution. Now, one of the things I was interested in is looking at the interaction between pollution and parasitism. And the main reason I was interested in this was because I thought it would be interesting to use parasites as indicators of polluted areas. The logic being that if you pollute an area, the fish in that area get polluted, and you see more parasitism in these stressed out fish because their immune system is affected, or they're not feeding well and they're weak, and so they get more parasites. So the hypothesis was entirely based around this idea that we would see more parasites in fish from polluted areas. So we had this great system in New Brunswick that we could work with. There was this beautiful river system where there was it was something called the Heath Steel Mine, which had been a gold mine and was leaching metals and other contaminants into the adjacent river. And so the contaminated water would flow down from this area here, down into the main stem of a river here, and downwards towards the estuary here. And it was great. It was a great system because there were adjacent rivers flowing in like this that were completely unaffected by the mine at this level here and at this level here. So we had multiple control sites that sort of gave us an idea of what a healthy, unpolluted system should look like. And then we had this theoretically polluted system. So the hypothesis was that the fish in this red zone here were affected by the mine and would therefore be more affected by parasites. And the fish in the green zones here would be less affected by the parasites. Again, I said about how my undergraduate research career got me used to failure, and it's a good thing because my hypothesis for this experiment was completely wrong. And in fact, the fish through here had lots of parasites on their gills. The fish in this red zone had few to none. And in fact, the closer you got to the mining site, the fewer parasites you found. I never found a single one of these gillworms at this site here, which is immediately adjacent to the mine, right? So, I got really cool results. We published these data and so on, really, really interesting. But it was the outcome that we got was completely unexpected. Compared to what we'd gone in sort of expecting, and this is something I've learned over time is a very common occurrence in scientific research. It's something you need to get used to, and in fact, I've learned to start celebrating it. That the unexpected result is actually cooler than the expected one. But it is important now to sort of sit back at this point of the talk and remind you that my early career was the place where my great hypotheses went to die. I had this idea that parasites cause growth stunting in pike, that got shot down. I had this idea that parasites and pollutants decreased the condition and meat quality in Arctic fishes, that got shot down. I had these ideas that parasitism would increase in fish exposed to pollution, and that got shot down. So I had some time after my PhD to sort of think about this, and I hooked up with probably I would refer to him as my top mentor, the person who's played the greatest role in shaping my scientific thinking and my development and overall attitude to science. And it's the guy in the middle of this image here, this guy right here. His name is Dave Marcogliese. He's now retired. He was a scientist with Environment Canada, and he got me thinking about parasites completely differently. And ever since then, my whole approach to doing research has changed. For example, one of the things that I started doing with Dave was looking at the effects of pollutants on parasites, and trying to figure out if the pattern that I saw in this system here was a generalizable one in parasites. And as it turns out, the answer is yes. Oh, a positive result. So it turns out that if you do a meta-analysis, in other words, if you go and look at all the published accounts of the effects of pollutants on parasites and the interaction of the two, you realize that consistently the effect size of the pollution on the parasite is negative. In other words, pollution has negative effects on parasitism, and it's consistent across the board. There are some situations where the data were a bit more sort of even, but consistently across the board, I was finding negative effects of pollution on parasitism. And so we published that back in 2009. And the publication of this paper was part of an awakening for me, a sort of another moment where I realized that I needed to stop thinking about parasites as being bad in a system and start thinking of them as being necessary parts of ecosystems that are damaged or harmed when those ecosystems get polluted. I'm not the only one who's had to come around to this change of thinking slowly. If you think about how parasites are defined, I haven't even defined parasitism yet. But parasites are effectively organisms that live on or inside other organisms for an extended period of time and extract resources from those organisms and cause them harm. And that last part is really important about how we define parasitism. This is a whole bunch of definitions of parasite that I plugged into a word map generator. And I've highlighted the harm words in red. And you can see how important these words are for how we define parasitisms. Parasites are defined as parasites because, in theory, they cause damage to their hosts. They harm them, they feed off them, they affect them negatively, they cause disease, they exploit them at the host's expense, yada, yada, right? If you define an entire group of organisms based on the harm that they cause, it affects your thinking about those organisms in the sense that you start thinking of those organisms exclusively as disease-causing organisms and not as the organisms that are just normal, healthy part of the larger ecosystem. And so over time, one of the big changes of thinking that have happened to me, and I think it's happened to a lot of other parasitologists, some caught on sooner than I did and have been talking about this for a lot longer than I have. But one of the big changes of thinking in parasitology in my profession has been this idea that parasites shouldn't just be viewed as agents of disease, but should be viewed as normal parts of healthy ecosystems. Now, that's not to say that parasites don't cause terrible disease and like things like malaria, we should study how to cure it or how to prevent it and potentially even how to eradicate it. But on the whole, parasites considered on the whole are probably nowhere near as harmful as we think they are. And in fact, the presence of parasites is probably a hallmark of a healthy ecosystem. And so there have been a number of papers that have tried to sort of promote this idea. There's the one on the left there, is a healthy ecosystem, one that is rich in parasites, and the one by my mentor, Dave Marcogliese, parasites of the superorganism, are they indicators of ecosystem health? In other words, does the presence of parasites tell you that that particular ecosystem is in fact healthy, right? It's weird to think that you'd go into a system and sort of find a diseased fish and say, oh, healthy ecosystem. But that's in fact, the more we study these systems, the more we've realized now that that is in fact the way you should be thinking. And in fact, I pull out a particular quote from Dave's paper here, which is the challenge for parasitology is to convince resource managers and fellow scientists, and in fact, the general public, that parasites are natural parts of all ecosystems and that healthy ecosystems have healthy parasites. And this has been one of the challenges for me, for other parasitologists, for the society that I'm part of, which is the American Society of Parasitologists. We've been beating this drum quite a bit to try to change people's perspectives on parasites, to get more students into parasitology, to study them as interesting organisms in their own right, rather than simply studying them with a view to eradicate them, if that makes any sense. And we're getting there slowly but surely. I like to think the perspectives in the past 20 years have changed quite a bit. So I did 3 years of postdoc at Environment Canada, and now I find myself here at Nova Southeastern. I've been here for 10 years now, and in that time, I've had a chance to develop absolutely fantastic research interactions with some of the best people I've ever worked with. I have ongoing research collaborations with great people who've actually spoken in this session here. People like Dave Kerstetter, who does great work on birds and fishes and is an ongoing Maine research collaborator. People like Amy Hirons, and Rob Smith. I've got starting up research projects with Lauren Nadler. And of course, I've got ongoing research collaborations with people outside of Nova Southeastern. I've got colleagues at University of Puerto Rico, at the University of Georgia, colleagues at University of Alabama. I've got ongoing people up in Canada, people up in Colorado, and so on. One of the great parts about doing this kind of work is that it fits in with so many different types of research. And so for someone with a short attention span like me, it's great to be working in a field that changes so much. So I've done work on freshwater and marine fishes, birds, including raptors, herons, pigeons. I've done work that focuses on invasive species like lionfishes and iguanas. And if you have any questions about potential research projects and so on, I'd be happy to answer them at the end of this talk. There is one type of research I want to focus in on, because it really highlights how bonkers and unexpected your journey, your adventures in parasitism can be. All right, so I'm going to finish with this case study, and then I'll take questions. If you've been anywhere near Florida, you know that the place is overrun with iguanas, and the place is packed to the rafters with them. They are invasive. They don't have any real predators, and so they've basically taken over. And the main one that's present here is the green iguana, or iguana, but there are two other species as well. What I'm going to say is based on research on the green iguana, but I'm sure it's true of the other two as well. Now, iguanas, every single iguana that we've ever opened up, and this is work that I've largely done with Dave Kerstetter, but we're starting to involve other people as well. Every single iguana we've ever opened up is infected with a parasite called Ozylimus megatyphlon. That's this word right here. And so this is the cecum, which is a branch of the intestine of freshly dissected iguana. And you could see that it's absolutely packed with worms. I don't know if you can see, but the entire volume is absolutely packed. This dark stuff here is partially digested food. So this is like a partially digested cocoa plum here. But this is, all of these worms are alive in this. And as I'm sectioning through, I'm exposing more and more of the worms. Now, this nematode, so it's a roundworm. As a roundworm, it's related to things like pinworms and hookworms and heartworms. These are things that you may have heard of that affect children and pets and stuff like that. But this is something that you find in iguanas. And every iguana I've ever opened up has these things in them, and has a lot of them. I mean, thousands of them, tens of thousands in the larger iguana. And if you look at the literature, the veterinarian literature considers this thing to be a parasite, not just a parasite, but a pathogen. And if you read the actual argument that they're making, they're saying that we found this iguana, it died in captivity. When we opened it up, it had tens of thousands of these parasites. Therefore, this parasite must have contributed to the death of this iguana, and therefore this parasite is a parasite because it's harmful and is a pathogen. And I think you could probably see the flaw with that reasoning. You can't just look at a dead thing and say, open it up, find a parasite, and say, aha, the parasite killed it. You have to look at a bunch of perfectly healthy iguanas, and if you still find the parasite in great numbers, maybe it's time to rethink your idea that this parasite is all that pathogenic. Anyway, so I thought this parasite was cool, and we had a bunch of students looking at it. And at one point, one of our students was looking at it under the microscope and noticed something weird about it. And so this is a composite image that we made of one that we pulled out of an iguana. So this is what it looks like. And so this is a bunch of images that we put together in PowerPoint here. And I think you could see it here. This isn't dirt. All right. This fuzziness here isn't dirt. It looks like the worm is hairy. All right. So we zoomed in a little bit closer and we saw this. Hairs sticking out of the cuticle of these nematodes, out of the skin of these nematodes. Now, there's no way for you to know this unless you're a parasitologist, but nematodes aren't hairy. They don't have this. This is not something that is a feature of nematodes. They have bits that stick out and stuff like that, but they don't have hair. This is not a feature that you see in nematodes. And yet, every single Ozylimus that we looked at had these hairs. And we were like, what is going on here? What is going on here? So we did some preliminary things. We thought, is it like a fungus growing on the surface? Is it maybe bacterial? And we thought, let's throw these things under a scanning electron microscope and see what they look like. So now here are some scanning electron microscope photos, and you can see this is a worm here, and you can see this whole patchy area here. This is the bit that's hairy. Let's zoom in and look at these hairs more closely. And if you look at these hairs more closely, you see that they form these dense mats on the surface of the cuticle of the worm. Let's zoom in a little bit more closely. This is not just some random thing growing on or on the surface of the nematode. This is a microbiome that involves filamentous bacteria that are actually embedded into the cuticle of these nematodes. And then they're sort of the forest. and then sort of, or the mat of bacteria, and then associated with that mat, you see an entire micro community, an entire microbiome living on the surface of these nematodes. So these ring type structures are the skin of the nematode itself, these sort of rings. So these are the folds in the cuticle of the nematode. These long spaghetti things are the filamentous bacteria. And then all of these smaller bits and pieces, these are all bacteria growing as part of that microbiome. So that this is a close-up of this here. And you can see all the basic bacterial types are here. You've got spherical cells, diplococcus type cells, you've got rods, you've got the filamentous bacteria growing here. It was absolutely bonkers. And so we were like, well, what does this mean for the nematode, and how does this compare? Maybe this is just the same microbiome that you see on the host's intestinal lining, and it's just colonized the surface of the worms that live inside the intestine. So we had this idea that maybe the worms were being colonized by the intestinal biota or microfauna of the, or microflora of the iguana. So we did, we extracted, this is, we did scanning electron microscopy of the host's intestinal wall, and you can see that there's no sign of a really well-developed micro bacterial community at all. So it's very, very different from what you see on the worms. So we did molecular analysis after this. Oh yeah, so the key result was that the nematode cuticle microbiomes are concentrated among these mats of filamentous bacteria, and the mats are not on the host epithelium at all. And so we did molecular work to sort of compare them. And on the left, you have the community of bacteria that are on the nematode. On the left, you have the nematode, the roundworm community, and on the right, you have the iguana gut community. And you can see that they're really different. This is a heat map that shows you how much these different groups are present. These different bacterial groups are present in different samples. And like over here, you see hotspots of bacteria that are typical of the nematode cuticle. You don't find them very much on the host at all. And you have a whole bunch of things here that are very typical of the iguana gut that you don't find on the nematode at all. So the iguana gut and the nematode have completely different microbiomes on their surfaces. So we looked at it a little bit more closely, and we confirmed it. The iguana gut samples and the nematode samples have very, very different communities. Now, if you look at these communities, and not only are they distinct, but the communities of microorganisms on the surface of the nematode are all plant cellulose fermenters, almost all plant cellulose fermenters, which means that the nematode is covered with exactly the right kind of bacteria to help digest the plant material that is the main diet of the iguana host. And there's actually more of them on the surface of the iguana than there is on the surface of the host's own intestine, which makes us think that maybe the presence of these worms on the surface or in the cecum is actually, I'm just going to go back here so you can see them now and how they're packed. Is it possible that the presence of these worms is actually beneficial to the iguana because the microbiome associated with these worms drastically increases the efficiency of digestion of this plant material that is the iguana's diet? So that's the next step for us, is that we're going to take this project to the next step, and we're going to actually see, we're going to play with this with the nematode community, see if we can remove the nematodes and see if that affects the condition and growth and development of the iguanas. And we've got a few other sort of projects to do. For instance, we haven't identified the bacteria, the filamentous bacteria, and so on. But what a bonkers thing that these parasites might actually be helpful to the iguana rather than being parasites. And if they are helpful to the iguana, are they even parasites, or are they mutualists? They're mutualists. If they're not causing harm, they have to be mutualists by definition. That's how we define the relationship. So are these things that we have traditionally thought of as parasites actually mutualists, in the same way that the gut microbiota of termites help them digest wood? There's a similar thing that goes on with kangaroos. The kangaroos have a roundworm that helps them digest their food, apparently. So it's like we've got all these things that we used to traditionally think of as parasites that may not in fact be parasites at all. And this is what I mean by unexpected relationships. I went into this sort of expecting to think about maybe this parasite can be used as a biocontrol strategy for the iguanas. And I've gone from thinking that to thinking, holy smokes, these nematodes may actually be necessary for iguana survival, which raises interesting possibilities for control of iguanas as well. We could talk about that in the question and answer period. So I really do want to leave enough time for questions. So I'm going to close off this discussion here just with a few closing thoughts. For those of you who are pondering research, who are either at the undergraduate stage or the graduate stage, or are pondering graduate studies and so on, the only thing I could say is that there's no such thing as the right path or the right type of path. I followed a very nonlinear path. I kept falling into parasitology essentially by accident, and it's ended up working out great. I know other people who had really clear ideas. Like I feel like my colleague Rob Smith. I feel like Rob had really clear ideas about what he wanted to do, and he's just made that happen, right? I know that Dave Kerstetter, my main research partner, has followed a nonlinear path like me, and so on. So don't think your path needs to look a certain way just because someone gets up here and gives a talk and says this is my path. Don't think that you need to follow that model. Follow your own model. Your own model will get you there. Be willing to follow the path of most fun. Just because something is fun doesn't mean it isn't serious. And just because something is fun doesn't mean it's not going to lead to really interesting and scientifically productive avenues. And the last thing I'll say is failure is good. Having your pet ideas thrown out, having your pet hypotheses proven wrong is not only is it a healthy part of science, but it actually tends to lead to completely new ways of thinking about systems, and it tends to be the more interesting result and the more productive result scientifically. So learn to embrace failure. You know what they say about fail? It's the first attempt in learning, and FAIL. And so I encourage you to take that attitude, because I have failed a lot, or rather a lot of my ideas that I thought were really clever have ended up being not so clever after all, and that's just fine. So I think I'll stop there, and I'll stop sharing my screen now, and I'll open it up to questions. I think that gives us about 45 minutes, it's about 15 minutes or so to take questions from the audience. So thank you very much for your attention. I really appreciate you spending time with me tonight, listening to me go on about my experiences, and I hope it was informative.
Dr. Blanar, first off, I want to thank you. That was awesome. I mean, I was literally over here. I was followed every word, and I'm not a science guy, right? Who's guys call me as non-science, but yeah, I think it was incredibly fascinating. We've got an awesome group of students and other faculty and other people that are engaged with the non-NSU community. So I want to open up the questions. There is a couple questions in the Q&A. I don't even know if you want me to read those, or you want to jump in and read them.
I could do that. I've just opened it up. Antonella. So yeah, it's I'm really happy that some of my graduate students got to join in. I don't know what I was thinking. I didn't notify anyone in my lab. I don't know what I was thinking. I didn't notify any of my students. I'm currently teaching a parasitology class. I didn't tell them until super late. Anyway, I totally dropped the ball on this, but I'm really glad that Antonella was on the ball enough. So Antonella is one of my grad students, so thank you for being here. Is it possible for a parasite to be considered mutualistic and parasitic at the same time? I would say that under most traditional definitions of parasitism, the answer is no. That the fundamental way that we define parasitism is harm to the host, or at best neutrality to the host. If you can demonstrate a benefit to the host, that is also, and that there's a benefit occurring to the parasite as well, it stops being a parasitic relationship and it starts being a mutualistic one. I mean, all of the relationships that we think of as symbiotic mutualisms have some extraction going on, right? If you think about rhizobium in the roots of legumes, the rhizobium extracts sugar from the plant. And you think, okay, well, that's extractive, but the rhizobium is giving is fixing nitrogen from the environment and giving back, I think it's a nitrate or nitrite to the plant. And so because there's a quid pro quo, it stops being exploitation and starts being mutualism. So I don't think something can be mutualistic and parasitic at the same time. And Tanella asks a follow-up question, and I'll get to Alexia's question. How do I think the classification parasite can affect future research in terms of mutualistic beneficial characteristics of parasites? Do you think the rest of the world will listen if we can demonstrate that parasites are not always harmful? Oh, you've just picked at a bit of a scab, Antonella. Yeah, and we walk this line in parasitology because a major appeal, it's not the major, well, it might be the major appeal, but a major appeal of parasitology is kind of the morbid fascination with things that cause disease. And part of the appeal is that these things cause disease, and or it could also be that you hate disease and you want to improve the human condition by finding ways of treating these diseases and so on. But the emphasis on the disease part brings us funding, and it brings us students, and it gets us attention at conferences, and it allows us to publish papers, because whenever you publish a paper, the very first thing a reviewer will ask is the who cares question that I got from my supervisor way back when. And part of the reason you might want to care about the parasite is that it causes disease and it might kill you, or it might cause harm and so on. And so the one of the ways that we've sold parasitology to funding agencies and reviewers and students and so on is by perhaps overselling the harm part. I like to think that as we sort of move towards thinking about parasites as important components of ecosystems, as things that even should be conserved, that are worthy of conservation, save the tapeworms and all that. I like to think that that people will still come, that people will still show up and be interested. I think it's up to us to do a good sell job and not just always talk about how negative and gross these things are, but talk about how beneficial they can be as well. Alexia, you said, did you find any negative impacts to fishes when the black spot disease was present? It's extremely common in sunfishes and bluegills. So Alexia, yeah, I grew up in like Quebec, and it's this parasite is everywhere there. As far as I know, the only studies that have found anything have found it in really, really, really heavily infected fishes, and in terms of their overwintering success. So like during the winter, when they stop feeding very often because either it's too cold or the food isn't abundant, or there's ice cover and they sort of hibernate underneath the ice, in the water underneath the ice. During that starvation period, apparently the most heavily infected fishes can sometimes die off. But there's no experimental evidence for it as far as I know. Some people looked at like thermal tolerance, and other people looked at swimming performance and so on. And surprisingly, even a fish like as infected as the one I showed you in that picture, which had thousands of cysts, it's very difficult to detect any effect. It's gross. Like the main issue, of course, is that it's gross and nobody wants to eat the fishes, right? Like I think that's one of the things about those fish is nobody wants to eat them. So aesthetically, it's largely an aesthetic problem, I think. Kate asks, can a parasite mutate and or evolve based on changes of migration patterns of the host? I don't know about mutate, but evolve definitely. Absolutely. Yeah, there's an entire really productive, really interesting area of parasitology, looking at how host behavior such as migration and changes in migratory patterns and so on affect their relationship with their parasites, and how that affects the host parasite relationship and host parasite coevolutionary patterns and so on. So yeah, that's a great question.
Dr. Blanar, we also have a couple in the chat section here. Kendra said, do you think the bacteria on the nematode would have been discovered if the student had never questioned it?
Absolutely. Oh yeah. No, this is 100% the student noticing it. I think had I seen it, I would have thought it was gunk on the slide. But this student said, hey, did you notice that nematodes are hairy? And I was like, what are you talking about? And they're like, yeah, no, look at the hairs. And the student happened to have focused in at a magnification where it was very clearly not like just fungal contamination or whatever. It was very clearly something that was attached to the cuticle. And I was like, oh. So I spent a couple of days reading about hairy nematodes and so on. And that's the very first paper I got was about nematodes in marine sediments that have symbiotic bacteria on their surface that help them process the chemical environment and get food from that environment. And I was like, whoa, what if that's happening inside the iguanas? And the rest is history. But it was all the student. It was all the student who noticed it. I like to think I might have noticed eventually, but given my long history of failure, I can't be too sure. Question from Emily, another one of my graduate students, by the way. I'm so glad that you were able to join it. I'm sorry I suck at telling you about this stuff. Regarding the research you did on gill parasites around the polluted mine, do you think there are widespread effects on the entire chain of species that are infected with these parasites? And could that be translated to different species hosts in different environments? Yeah, absolutely. So the gill parasites themselves really only, in that particular system, really only infect the Atlantic salmon that are in that system. But I am told that the free swimming form might be grazed on by other things. And so that might be those organisms that graze on the parasites during their brief free living phase, that they might have been negatively affected. But really, it's more about what that lack of parasites in that system tells you about more what's going on in that system in general. Like it tells you that system is being altered in a pretty fundamental way. If you're looking at the fish and you're finding that there's not a single active parasite on any of those fishes, that's weird. You know, once you've worked with fish for a while, like not finding any ectoparasites at all is very, very strange. I'll go into the chat for a second. I'll just look at Elizabeth Warburton, one of my collaborators. We just got a paper out. I'm so excited to get it out. Hey, Chris, nice talk. And those pike were really small. Yeah, but kind of technical question we can talk about later. Ooh, let me look at this. I wanted to ask about the nematode bacteria association. Some helminths have been found to receive developmental cues from the host microbiome that let them know they're in an appropriate environment. Could you speculate if that could be somewhat related to the iguana nematode bacterial association that you're seeing? Oh yeah, we need to talk. This is a great idea. Yeah, let's talk. Let's talk about this because that's kind of a cool idea. And that could be a separate experiment. I'm going to copy and paste this question. Liz, I'm stealing your idea. Copying and pasting it into another document. In the chat, hold on a second. Let me move this out of the way. Kendra, do you think the bacteria in the nematode would have been discovered? Oh, so, yeah, Kendra, I just answered your question. Absolutely, full credit goes to that student. I'm not sure I would have spotted them, and in fact, I think it's a pretty safe bet I would have missed it. And Eben, hi Eben, a potential research collaborator, one I'm looking forward to working with very much. If I run out of student questions, can I unpack the difference between effects of direct and indirect exposure on parasite richness from the Cohen's D plot that I had? Let me see if I could call this up quickly and share it. I'm getting a lot more technical questions than I thought I would. This is a smart crowd. I can tell.
It is, but don't worry about time though. Too, Dr. Blanar, if you want to answer your questions, like it's not going to shut off right at 7.
Okay, good. Yeah, I've been looking very nervously at my watch, and I see like it's 6:57. So, Eben, your question is about type of exposure, indirect and direct. 2009, Eben. I'm going to have to access my brain vault for this one. So the level of organization, population, community, that's pretty straightforward. Life cycle complexity, so single host versus multi-host, type of exposure, indirect versus direct. I think what we were doing there was looking at whether the paper that had yielded that data point or those data points involved direct exposure of either an ectoparasite or the free swimming stage of an endoparasite versus indirect exposure. In other words, it's the host that's swimming around in a polluted environment, and the parasites inside that host are presumably being indirectly exposed. That's my recollection of what we meant with that. Feel free to ask follow-up question if that doesn't make any sense. But that's my best recollection of what was going on in that. Antonella has asked another follow-up question. One more follow-up question. If we sell the disease and morbidity aspect of parasitology, and we're also trying to sell the conservation and beneficial aspects of parasitology, what do you think of the future of helminthic therapy? Wow, that's a really good question. I think that the future of helminthic therapy, or the development of helminthic therapy, has been strongly held back by attitudes towards parasites and concern and the ick factor, for sure. In the same way that like maggot therapy, which is a thing, and those of you who haven't heard of it should look it up. But maggot therapy is a thing, and it's being held back by the ick factor. Leeches are being used in surgical applications for like things like reattachment surgery. But the universal application of attaching leeches to wounds and stuff like that is still sort of being held back by the ick factor and so on. So yeah, we're kind of in a catch-22 because we play up on the ick factor to get funding and to put bums in seats and to sell talks and stuff like that. And we have an entire show called Monsters Inside Me, which is all about parasites, and we emphasize the monster part. And the whole show is set up like a horror movie, like with weird lighting and like dramatic sort of like sound effects and stuff like that. Yeah, so I think that the future of helminthic therapy depends on our ability to, well, first of all, demonstrate that it's effective, which I'm not sure that that's been done yet. But also, even if we demonstrate that it's effective, getting past the ick factor, and that you could do it in a controlled sort of way and administer the parasites in a controlled way, minimize transmission to people that are not the patient, or to the larger community, and so on. If you could demonstrate that, then I think that'll be part of the victory won. I mean, my big question is, do you think people would be more receptive to an RNA vaccine or being administered 300 whipworm eggs? I don't know. One is natural; it's 100% natural, and the other one is is what it is. I wonder if people's attitudes would be different towards, that could be an interesting sort of survey type study to do with an undergraduate, perhaps interested in social sciences or something like that, or perhaps a graduate study, because if you do it right, if you make the scale appropriate, those of you who might be considering a social science type thing or are interested in these type of survey-based projects, that could be an interesting idea. The public attitudes towards parasitism and the future of helminthic therapy, I think would make a cool project for someone. So
I think that's all the questions we have. Anybody else have any additional follow-up questions before we wrap it up? We do want to thank you, Dr. Blanar. This was an awesome lecture. I know I said that earlier, and I also appreciate everybody who attended tonight, was interested in the topic. And there'll be some more virtual lectures coming up forward. We'll probably end up connecting again here. You'll probably see some other lectures. There's a couple coming up from some of our current graduate students too, as well, over the next couple of weeks.
Well, I want to thank you for inviting me to be part of this. This was great fun to put together. It's definitely unlike any talk I've ever presented because of the audience and because of the types of sort of cues you gave me. And so I've had a lot of fun doing this. So thank you very much. And yeah, and if anyone, oh, there's Devin as well. I do. Yeah, so if anyone has any questions about like projects and ideas and stuff like that, I am always happy. You can get my contact information through Matt or through the university websites. If you just Google me, you can find me.
We send everybody in the audience just a follow-up email, just on, if you don't mind, we'll share your email or contact information, Dr. Blanar, and if they have any questions, they can follow up with you.
Yeah, thank you all for attending. It's taking time out of your busy schedules. I know how busy we all are. I see a lot of like colleagues and stuff like that in the attendance list. I'm absolutely blown away and honored that you took the time to sit through this. And I hope I didn't talk too much nonsense out here.
No, it was great. Thanks, everybody. Have a great night, and we'll connect again. Bye.